prokaryotic and eukaryotic cells. Prokaryotic cells are smaller. They have cell wall, which is made up of peptidoglycan, right? And they are present in bacteria alone. You won't find a single prokaryotic cell in animals, plants, or higher organisms in general. Okay, what is the main difference? That DNA is present naked in the cytoplasm and it is not covered by nuclear membrane. Prokaryotic is taken from a term pro, which means before, and karyos, which means nucleus. So it actually means prokaryotic cells were originated before even the origin of nucleus. They came before nucleus, right? So that's why the DNA here is circular DNA, which is double-stranded, of course, but it is lying there in the cytoplasm without any covering. There is a slime capsule over it. Then there is a cell wall made up of peptidoglycan. And finally, the third innermost structure is the cell membrane, which is made up of phospholipids. Plasmid are extra chromosomal DNA. They're also circular, but they're much smaller than the main bacterial chromosome. Flagella can also be there, which is used for locomotion. Now, the relative size of, this is what I have not discussed, the order of magnitude, relative sizes of the cells with respect to each other. Here is a scale given. You see, a small molecule can be as small as 1 nanometer, a very small molecule. DNA is around 10 nanometer. Virus is 100 nanometer. But there are viruses which are even smaller than 100 nanometer, like a virus which causes common cold, a rhinovirus, or poliovirus. You might have heard about them. They're just 30 nanometer in size. It's a bacteriophage, which is 100. A prokaryotic cell is just 1 micron, and eukaryotic cell may range between 10 to 100 micron. Okay, so now what is what? 1 nanometer is equal to 10 to the power minus 9 meters. 1 micrometer is equal to 10 to the power minus 6 meter. Okay, then the size of a millimeter, an ant is millimeter. Millimeter onwards, you can see things. It's all in the macro scale. So you see, you will be able to see them through your eye, and rest of them for observation, you need electron microscope. Now, prokaryotic cells, till wasp, you can even observe them using the light microscope. Correct? Done. Now, specialization in animal cells, we discussed briefly. We talked about different special cells in animals, like the nerve cells, which is like, having a characteristic shape and the function of it, like dendrites will, carry the impulse. And the impulse will reach the cell body where it is processed and then goes to the axon and then to the terminal where it is released in order to transfer the signals to the next neuron or to the muscle cells. Muscle cells are another variant. You have two types of muscles, voluntary action that is dependent on your wish. It may be striated, striped, skeletal muscles. Non-voluntary action ke liye you have smooth, unstriped, unstriated muscles. Then the sperm cell, which is also different than the regular cells because it has got a head containing acrosome. This contains digestive enzyme, which is needed to penetrate through the barriers of egg cell. Mitochondria is present in the middle piece. This is known as middle piece, which is rich in mitochondria because they need to produce a lot of energy and give it to the flagella present in the tail region so that the tail can help the sperm swim through the spaces. Right, moving on to the specialization in plant cells. We have root hair cells, which are specialized. Specialized for what reason? They need to absorb lots of water and the dissolved mineral ions. So they are specialized in such a way that the uptake of water and mineral ions can be facilitated. Okay, they are always relatively close to the xylem tissue because xylem, the root hair cells will be absorbing it and transferring it to what? To the xylem tissue, which will carry it upwards. Right.
Okay.
The xylem tissue carries water and minerals up to the rest of plant. Mineral ions are also moved into the root hair by active transport. What is this active transport? We'll discuss that in a while. So root hair cells have three main adaptations. One is that it greatly increases the surface area available for water to move into the cell. Wherever this term absorption comes, absorption of whatever, you will have the absorbing structure actually enhances the surface area for maximum absorption. The same thing we discussed in human digestive system as well, that villi are the foldings which increases the surface area. And there is also presence of a large permanent vacuole that speeds up the movement of water by osmosis from soil across the root hair cell. And they have many mitochondria which transfers the energy needed for active transport of mineral ions into the root hair cells. So what is this? Why active transport? Why the energy is needed? Why shall they have mitochondria? We'll be discussing in short while. You have nucleus here in a root hair cell, a large permanent vacuole, mitochondria can be seen, and root hairs are here. So what exactly happens is when water is needed to be taken up by them, water will move, water will be absorbed along with the mineral ions. Like their transport is a bit different. Try and understand. I'm explaining transport and absorption of water and mineral ions right here. there are two basic types of transport in living cells. One is the passive transport and another is active transport.
Yeah.
And second is active transport. Now, in passive transport, energy is in the form of ATP is not needed. So we'll write ATP is not needed.
Okay.
So how exactly the transport is taking place? It is either by simple diffusion, there are two types, simple diffusion or facilitated diffusion. Or facilitated diffusion. Active transport is where ATP is required.
Yeah.
And the movement of molecule is against the concentration gradient. These are the 2
Sure.
points. Against the concentration gradient. In case of passive transport, both the diffusion will not require, will be down, or you can say along, down or along the concentration gradient. What does that mean? It means that, let's show here. This is simple diffusion here. Simple diffusion. In case of simple diffusion, we have, let's say this is a barrier. What is this barrier? The cell membrane. Now, there is some small simple molecule. You may consider that a gas, carbon dioxide. Or let's say, because it's coming out to in, let's say this is oxygen. Oxygen moves from a region of higher concentration. The concentration is high here. The concentration is low here. You see, it is moving down the concentration gradient. That's simple diffusion. No energy needed. The molecules will be simply diffusing, moving from one area, from a region of higher concentration to the region of lower concentration. Clear? Facilitated
Yes.
diffusion. Facilitated diffusion is where it's not simple. It needs to be facilitated. Why and when? Sometimes, the molecules are not as small as oxygen. It's not oxygen or carbon dioxide gas or some iron. It's actually, let's say, glucose. Glucose cannot just simply diffuse through the membrane. Even if the concentration gradient at one side, concentration is very high and the concentration is very low, the other side. Even in those cases, it won't be moving simply. Why? Because the structure is big. There's no simple movement. Thus, it is a bulky structure. Plus, glucose is polar. Glucose, or let's write, it's not just about glucose. So we'll say that the polar and bulkier. What is the meaning of polar?
Polar.
Yes.
Is it like two-way, like or different?
Polar means that it is water soluble. It gets dissolved in water. Okay. So polar substances and those which are bulky, they're big. They cannot pass through the membrane without the help of, I'll have to make it again, without the help of some transport protein. They can only pass through these transport proteins because they'll make a channel which will allow or accept the polar molecules to pass through the membrane. But then it will again be down the concentration gradient for sure, without the need of energy, without the need of ATP.